Okay, we are going to draw cellular respiration. So I'm going to start by drawing the cellular membrane, which, if you remember, is a phospholipid bilayer, like so. Okay, so this would be the cell membrane of a cell that would be gigantic. It would stretch up and down and out at least, I don't know, 10 feet. Okay, and then every cell is in close proximity to some kind of capillary or blood vessel. It's usually a capillary. It's about the size of a red blood cell. So you can see here I'm going to draw a red blood cell. That's a little divot in the middle. And they're flowing through this capillary really close to this particular cell. Oh, and I've left a gap in the membrane because that represents our GLUT4 receptor that we learned about. Okay, this is GLUT4. You learned about that when we were talking about diabetes, and so it's a facilitated diffusion receptor. So you learned about GLUT4 in your diabetes homework, and we'll talk about that again in a moment. Now, if you've just been eating, you will have glucose floating around in your cells, and I'm going to show it as a hexagon. Remember, it's a 6-carbon sugar, C6H12O6, and it's floating around in your cells. You've also just taken a breath, and so we will also have oxygen floating around in the cell. So I'm going to draw that now, and it's usually attached to a hemoglobin on a red blood cell. Okay, so let's say it's first thing in the morning, your cell's been starving, and so by simple diffusion alone, where does the glucose want to go? Well, it wants to go inside the cell, but because it's too big to pass through the membrane, it has to pass through GLUT4. Do you guys remember what that kind of transport is called? I'll just write it up here. Remember facilitated diffusion? Alrighty. Alright, so inside the cell, the organelle that's responsible for performing the cellular respiration is the mitochondria. So I'm going to draw it here. Now obviously this is not to scale. I can't really fit an entire mitochondria in the size that I've given myself, but we're going to make it right here. Now the mitochondria has an outer membrane, and then it has this folded inner membrane. Okay, and we'll talk about why it has folds in a minute. It facilitates a very important reaction in cellular respiration. Okay, and I've greatly increased the size. It's usually folded so much that it pinches in. And we're going to label these parts. So up here we have the outer membrane. And in here is the inner membrane, but it actually has a name. Let's see, we'll label it right up here. It's called the cristae. The cristae membrane. And then the space in between the two membranes is the intermembrane space. Scientists are not creative. And that becomes important because we'll see something happen there. And then the very inside is called the matrix. Okay, and then out here, this stuff out here would just be simply the cytosol or cytoplasm of the cell. Okay, so Let's say it's early in the morning, we're hungry, the glucose is going to diffuse by facilitated diffusion through GLUT4. And when I bring it in, I'm going to go ahead and open it up as a 6-carbon chain like we've been working with in our exploration activity. So there's our 6-carbon sugar. This is glucose. Okay, so the very first thing we did, you remember from the activity, is that we broke the middle bond. So we're going to break this bond right here. And by breaking that bond, first of all, is this an endergonic or exergonic reaction? It's an exergonic reaction, and so if we were to draw the reaction diagram, it goes like this. But notice that it goes up first, this little hump right here. That is the energy of activation. So it turns out we have to put a little bit of energy in order to get energy out. So we're going to take 2 ATP that are floating around somewhere in the body, and we're going to put that into the reaction. Okay, and that's going to get it started. And once we break it, we're going to get out things. Now remember, every time we break a bond, we're going to get 2 electrons out. And where do those electrons go? Well, they get temporarily stored on our storage molecule, which is NADH. So we'll draw that here. Let's see, I'm going to draw in orange. We've got our electrons coming out, and they're going to get stored on an NADH. So I'll draw that here. Okay, and then we also get a little bit of ATP out. So there's a little bit of extra energy when we break those bonds. Now this is where our analogy breaks down just a little bit, and it doesn't actually add up 1-to-1 like we did in our exploration activity, but that's okay, who's counting? So we get 4 ATP out, we put 2 ATP in. So you can see you get a net gain of ATP from this reaction. And then obviously the products of this are 2 3-carbon sugars. Okay, the 3-carbon sugars have a name. They're called pyruvate. Now, this process that we've just drawn, the breaking of glucose in half, is called glycolysis. I'm going to draw in black. Glyco, which means sugar, and lysis, which means to break. Glycolysis. Okay, we get 2 pyruvate molecules. I've just labeled one of them there. This ATP production is simply taking a P and loading it onto an ADP like I showed you in class. And so we call this substrate level phosphorylation. So I'm just going to draw that in there, where we just took it off of a substrate and we placed it on ADP. We phosphorylated it. Okay, we're going to see that type of ATP production one more time in this process. All right, so now we've broken the first bond. What do we need to do now? Well, obviously we need to break those other 2 bonds of each pyruvate. So the rest of this process happens inside the mitochondria. So on that note, if you have this happening in the cytosol, what other organisms might be able to perform this process absent a mitochondria? That's right, we've got bacteria that can do this, and that's what bacteria do. They just do glycolysis because they don't have the mitochondria. But in order to continue on and produce more energy for a larger body like ours, we need to get the rest of the energy out of there. Now would be a good time to pause your video. Okay, so we're going to take our pyruvates inside the mitochondria, and they simply go through as some kind of transport protein that we don't need to know for our classes. And here we go, pyruvate inside. All right, so we're going to start by breaking the next bond. So we're going to break this bond right here. Now we've got a single carbon. It's actually got some oxygens attached to it, so it becomes a CO2. And where does that CO2 go? Well, as the CO2 builds up inside the cell, it's going to passively diffuse through the membrane, back into the bloodstream, back onto the red blood cells, back to the heart, back to the lungs, and you exhale it when you take a breath. All right, so now we've got this 2-carbon sugar, and it's called an acetyl group. We've seen these before in histone acetylation. Acetyl is just the fancy name for 2 carbons. So there's our acetyl group. Now, we want to break that last bond, and it does happen in a process called the citric acid cycle. You've probably seen it as the Krebs cycle in maybe a past class in high school. And so I'm going to draw that here. We'll call it the citric acid cycle. And it's a cycle because we start and end with the same product. Okay, so there's a product that's already in the cycle. It's a 4-carbon sugar, which we won't name because it's not important. And it's going to get these 2 carbons added to it and then spit back out those 2 carbons and end up with the same 4 carbons again. The problem is that the acetyl group can't seem to make its way to the citric acid cycle all by itself. It's like a lost child, and so it needs a little bit of help. It needs a babysitter, if you will. And so we have a babysitter. It's a coenzyme, and it's the first one they characterize, so it's called coenzyme A. And for short, we just call it CoA. CoA. So CoA binds to the acetyl group, and this becomes acetyl-CoA. And the CoA is simply going to take it over to here, drop it off, and then go back for more. So it doesn't actually get involved in the reaction, it just brings the acetyl to the citric acid cycle. Okay, now would be a good time to pause your video and get caught up. Okay, so once the 2 carbons have gotten in there, we're going to break that final bond. Now this is where the math really doesn't add up with the analogy we used. There's a lot of reactions, 10 reactions that happen in the citric acid cycle of breaking and making bonds, and so there's a lot more product that comes out than we would think with one broken bond. But you are going to get those 2 carbon dioxides back out of the cycle, or the 2 carbons you put back in, and they come out as CO2. Okay, so the 2 carbons that we put in come out as CO2. I'm just going to put in that parentheses, it's CO2. And those are both going to exit the cell into the bloodstream on the same trajectory and be exhaled out of the body, just like the first one. The next thing that comes out of it are electrons because we've broken bonds. And so we're going to draw those here in orange. So electrons come out, and we simply store them like we've done before in a temporary bond between NAD and H. We actually get quite a few of them from this reaction, but we're not going to count. The last thing that comes out of this reaction is a little bit of ATP, again by substrate level phosphorylation. So we do get a tiny bit of ATP out. It's just 1 per turn of the cycle, so 2 per glucose molecule, because look, this is 1 pyruvate that has to go through, the second pyruvate has to go through as well. So 2 turns of the citric acid cycle per glucose molecule. And as I said before, this is also substrate level phosphorylation. This one and these. Okay. So now we have gone 2 turns of our pyruvates through the cycle. How many carbons do we have left? Well, none. They've all exited the cell as carbon dioxide, and we can count them, right? Here's 1 here, 2, 3, and then this whole process happens again, so 4, 5, 6. So all of our carbons are gone. The only thing we have left are these high energy electrons that we've stored here and here. Oh, and one more that I forgot. In this bond, when it breaks, we do get electrons out. So we would have stored those as well as an NADH. Okay, now would be a good time to pause the video and get yourself caught up. Okay, now that we have all these stored electrons, if you remember, these are high energy electrons. So I'm going to draw a diagram of them right here. This is our little atom. Okay, these are high energy electrons that are sitting way out here. Okay, because they're coming from a glucose molecule and they've gotten there by a plant through photosynthesis, which we'll talk about another time. And so we need to bring these high energy electrons down. Now remember, in combustion, they come all the way down to ground state in one foul swoop, and we get an explosion. That's not what we want inside the cell. And so we're going to do it more like fluorescence. So we're going to take them down 1 single layer at a time. And so to do that, we have what's called the electron transport chain. Okay, so I'm going to draw it along the Christie membrane. I'm going to label it over here. So this is now the electron transport chain. Okay, an electron transport chain consists of several proteins that are embedded in the membrane in the Christie membrane of the mitochondria. Okay, so I'm going to make some little spots for them. So you'd have a protein there, protein there, protein here. Now I don't care that you know how many proteins there are, it's not important. But there is a very important one down here which we'll talk about. It kind of looks like a fish. It's actually so much cooler than a fish. And this is our electron transport chain. So what happens is all the NADHs that are coming from over here, and here, and here, they all come over here and they drop off their electrons. So remember, NADH is just a temporary electron storage molecule, and so it's going to drop off all of these high energy electrons right here. They're going to be dropped onto this protein right there. Now this protein has them held in an outer shell, but each successive protein in the chain has one less electron shell. So I'll draw it here. So kind of, if we were to picture it, it would go like that, and like that, and like that, etc. And so as these electrons are being passed, they're falling a little bit in energy. Does that make sense? Okay, and so each time they transfer from one protein to another, a little bit of energy comes out. Now remember from our little analogy, we use that energy to do active transport. So every time the electron falls, we take a hydrogen ion, a little proton, and we shove it across the membrane. And it actually doesn't get past every single protein. That's really not that important. We do get a lot of passing of protons. And so you'll see that as all these electrons from the whole process get passed down, we end up with a situation like this, where we have a whole bunch of protons in the intermembrane space. So we've built up a proton gradient by using the electrons doing active transport. Now, at the bottom of the chain, the electrons, their final jump is onto an oxygen molecule. Okay, so where did the oxygen molecule come from? Well, it was in the bloodstream, and by passive diffusion, because we have high oxygen here, low oxygen here, it's going to make its way in, and it is logically going to go to the place where free electrons are being given out, since it's very electronegative. So oxygen is sitting right there at the bottom of the electron transport chain. In fact, it is the driving force for the electron transport chain. Because it's sitting here, it's almost like a vacuum, sucking those electrons down. So this one gets its electrons sucked away from it, so it sucks them from here, which sucks them from here, which sucks them from here, which sucks them from here. So each protein in the chain is progressively more electronegative, with oxygen being the most electronegative at the very bottom. So that last jump of the electron goes on to water. So now all of a sudden we have an oxygen. It's really just one oxygen molecule. We have an oxygen molecule, an oxygen atom, excuse me, that has 2 extra electrons. And so all these positive hydrogen ions are hanging out around there. So what do you suppose happens? Those 2 hydrogen ions are going to be attracted to that oxygen, turning it into water. Now we have all these protons, out here in the intermembrane space. And before we go further, it's probably a good time to pause and catch up on your drawing. Okay, so we have all these protons on the outside in this intermembrane space. So by simple diffusion alone, they would like to go back in, but because they are charged, they have a hard time passing through the membrane. And so we have a special transport protein, just like the one on your activity, that's going to allow them to come back through. And its name is ATP synthase. And it's probably the coolest protein on the planet. It actually has a spinning turbine head. So as the protons go through, it begins to spin. And as it spins, it uses the force to take a phosphate and an ADP and going around, slap them together to make ATP. So, and it makes a lot of ATP. As these go flying back through, the protein starts to spin and out pops ATP. Lots and lots of it. Okay, so where here you're getting 2 net ATP here, 2 net ATP here per glucose molecule. Here you get 32 to 34 ATP per glucose molecule. So this is a much more efficient, although very elaborate and complicated way of making ATP. Isn't evolution grand? And so this type of ATP production, instead of being substrate level, where we're just pulling a P off of something and putting it onto an ADP, we are using an oxygen-driven system to produce ATP by a physical force, the turning of that protein. And so it's called oxidative phosphorylation. And it's a much more efficient way of making ATP. Okay, so just to review the steps of cellular respiration, it starts with glycolysis. This is step 1. Okay, then it goes to the acetyl-CoA step, that's step 2. Then we have the citric acid cycle, which is step 3. Then the electron transport chain, which is step 4. Okay, so pause your video, go ahead and complete your drawing, and then we'll talk about what happens when oxygen is not present.